Yeah, perfect. Thank you very much. So yeah, today I'm going to talk about one of the most versatile protein folds in biology, that are the GNATs, and about their structural and functional evolution. So first of all, GNAT stands for GCN5 related and acetyltransferase, and it has a very conserved fold. And the process that it catalyzes is quite simple. It just takes acetyl-CoA molecule and it transfers that acetyl group to a substrate, to a broad range of substrates, producing the acetylated substrate. But it's an interesting protein because it's ubiquitous in all domains of life and it's involved in many different functions. Just to list a few, it's involved in bacterial antibiotic resistance, in chromatin remodeling, or in metabolism regulation. And the fact that it can acetylate such a broad range of substrates, we quickly saw it when we compared this superfamily, the GNAT superfamily, with all the other superfamilies we have analyzed in CAD. We quickly saw that it ranks amongst the most sequence and structure diverse superfamilies. And if we zoom in inside, we can see that at 35% sequence identity, we have many singletons that already show the huge divergence that this protein fold has been through. And here I'm highlighting in the center, we have a few clusters that are connected to each other. If they value between the clusters, it's lower than 10 to the minus 12. And I'm highlighting two different proteins, the human spermidine acetyltransferase and a protein from bacteria from Vibrio cholera that catalyzes the same reaction. And this is the question that our experimental collaborators came to us with, Misty Kun from the San Francisco State University. They had these two proteins that catalyze exactly the same reaction, but they look very different. The human variant, it's a domain swap dimer. You can see here the C-terminal length of the protein, this beta strand, and this alpha helix, how it swaps into the other domain, while the bacterial version, it's a dodecamer that doesn't domain swap. So we were wondering, how is it possible that even if we align the monomers, we just take one monomer of each of the structures, they superimpose pretty well, but they belong to so distant organism, such a different oligomeric state, but still catalyzing exactly the same reaction. So to better understand which are the evolutionary pathways that led to the function of these two different proteins, we performed a number of experimental and computational analyses. Our collaborators, of course, performed the experimental side of it. I focused on the residue and structure conservation. I mapped the substrates activities that they reported to us and tried to understand how the substrate recognition process works. And then, of course, we also focused on the different oligomeric states and how that is related to not only these two sequences, but also to the neighboring sequences. Because, of course, we don't only wanted to focus on these two entries, but also on the neighbor sequences in the sequence space that perform similar functions, that have a similar function, and that also share similar oligomeric states. So what we did to generate our starting data set was, starting from the CAD superfamily from GNATs, we took the two functional families that contain the human and the Vibrio cholera variant, performed a simple HMM search with a very loosey value of 10 to the minus 3 against UniProt, and we generated this combined data set that we call the PAT zone, because those are the proteins that are likely to have this polyamine acetyltransferase function. This led to over 30,000 different sequences, 37,000 different sequences that we had, of course, to narrow down to allow for experimental analysis. And to do so, we regenerated functional families for this extended data set, and we predicted structures for each of the representatives of those functional families, clustered them again based on structure, because we know it's quite important to define that oligomeric state. And for that, we use fold-seq and a TM score of 0.95. And with this pipeline, we ended up selecting 131 different representative structures that we combined with a set of 12 entries from which our collaborators had experimental data for, but that was not included in our PAT zone. Then, and it was mentioned, a similar problem in the presentation earlier, sometimes we cannot really align proteins just based on the sequence because it has diverged too much. For example, the human and the Vibrio cholera variants have a sequence similarity of the sequence identity of 10%, which is very low. So what we did was because we either had the experimental structures of those 143 representatives from PDB, or otherwise we predicted their AlphaFold 2 models. We used MTM-align to superimpose pairwise all those structures and obtained a structure-based sequence alignment. You can see here the RMSD values obtained of those pairwise superimpositions. And you can see that in most of those cases, it's below 4 Armstrong. So we obtain a good superimposition. Then using that structure-based sequence alignment, we used IQ3 to infer the maximum likelihood phylogenetic tree. And here we don't really need to go through all the sequences, of course, but you have Vibrio up there, the human one here. But there are a couple of interesting things to see already. You can see that there are some eukarya entries in very light blue that are intertwined with some bacterial entries, and we also have a clade with archaeal sequences here, if you can see. And notably, we have a single eukaryote entry that belongs to a fungi up there in the middle of all the bacterial sequences, which already started pointing out that there is an interesting evolutionary history behind this protein fold. So then what we did was selecting 71, so we get a good coverage of all the entries in the tree for experimental testing, from which 41 could be solubly expressed and screened against a set of substrates. Additionally, and to get some information on the different oligomeric states, we predicted the most likely oligomeric state of those 41 proteins. Of course, if we had experimental structures available, what we did was just infer it from the biological assembly, from PDB. But otherwise, the way we used AlphaFold3 was for each of those entries, predicting the dimer, tetramer, hexamer, and dodecamer. And the one that had the highest IPTM interface predicted template modeling score, we assumed it was the most likely oligomeric state, always that it was higher than 0.8, of course. And here we can also see that we have some areas that are quite rich in certain oligomeric states. For example, the domain swapped that I'm represented in pink here is the one characteristic for human. We also see it in a few bacterial entries down here, while, for example, the non-domain swap dimer or the dodecamers, we see them mostly around this area and linked to bacteria, but not exclusively, because we also see that lonely eukaryotic entry, that fungi, also seems to be a non-domain swapped dimer. So then we had to include the substrate information, the substrate screening that our collaborators performed, from which they selected 37 different known substrates for GNAT that just to simplify the downstream analysis, we grouped in four different clusters based on their chemical similarity, but I will not go to the detail on here. For this, you can see a few of the representative chemical structures there. And then we had to link it to the protein site, to the binding pocket. So because the domain swap process changes quite a bit the binding pocket and how it is shaped, it's difficult to compare structures with such different pockets. So what we did was taking 5 PDB structures that were co-crystallized with a substrate in the acceptor site of the protein, and we took all the residues that were within 5 Armstrongs of the substrate. It's just a classic procedure, and we selected the 12 positions that were present in at least 2 of those protein substrate complexes. And here I just mapped them to the human spermidine spermine acetyltransferase, and you can see more or less how they are distributed. We have a few that are part of the domain swapped region, and of course at this point we're only taking into account a monomer. We're not considering the positions that come from the other monomer that forms the domain swapped interaction. So then what we did was taking those 12 positions for all the 41 entries that were screened, checking the grand thumb distance. This essentially computes the similarity between the residues that we have in the binding pocket. And then we used that information to build that tree by building an all versus all matrix. And we added the substrate group as a pie chart in the notes to observe how the similarities in the residues in the binding pocket are reflected in the substrate group that those proteins are interacting with. You can see here, for example, that all these nodes mostly interact with substrates of the group 4, is the group of the spermidine. This is where we have the human entry. But again, and quite interestingly, we have the Vibrio entry up here, so with a very different pocket to the human one interacting with the same substrate group, as we already knew. So then, to take a deeper look at how those residues are conserved, we went to the 2 functional families that contain the human and Vibrio entries. You have the human one, the domain swapped on top, and the Vibrio entry at the bottom. And we compared we just generated a logo for those functional families, and we compared the positions that were exactly in the same structural location. So the first thing that we can see is that we only have 4 positions that are in the same location and that interact with the substrate. And if you check the conservation patterns, they are quite different. This leucine maybe is not that striking here, because then it's conserved to an isoleucine or to a phenylalanine. But if you compare this aspartic that is compared here to that is conserved in the Vibrio functional family to a glutamine, you can see that the way that this protein has evolved to interact with this substrate is completely different. Then, and luckily we had included 2 entries in our analysis that belong to the same species, to Pseudomonas aeruginosa, but that were quite different because one was domain swapped and the other one wasn't. We could see that we had one of the 2 entries here at the top that superimposes very well to the human domain swap dimer, while we have another one, also from Pseudomonas, that superimposes, that aligns pretty well to another eukaryotic entry. And this is that fungi, that isolated entry that I mentioned earlier in the top of the tree. And you can see that also the sequences identity, here it's 34%, here 22%. They are quite low, but the structural superimposition leaves no doubt that they have a shared ancestry. But when we compare the 2 Pseudomonas entries against each other, we get a 21.1 sequence identity percentage, so a bit lower than in these 2 different cases, which made us starting to suspect that maybe Luca, the last universal common ancestor, already had 2 copies, and that's why we're seeing that they behave in such a different way, that they have such a different oligomeric state, but still they have evolved to bind to the same substrate. So what we did to further verify this was using this nice piece of software, OrthoFinder, that what it takes is, it takes as input the proteins of the number of species you're interested in. Here, of course, we included the Homo sapiens and Vibrio cholera, as they are our main targets of this analysis. But we also included the eukaryotic entry, Aspergillus fumigatus, up here. We included Pseudomonas aeruginosa because of these 2 entries that are these 2 proteins that are quite different to each other. And then Bacillus, because we had it as an hexamer here. So we also wanted to take a look at this type of oligomeric state, and Anarchia, to have a representation of all the domains of life. These are a few metrics that OrthoFinder reports after running it. And this is the one that you normally need to check to be sure that it performed well, that it was able to build a species tree first, and that then will help on gaining confidence on the different orthogroups that it generates. Because this is what OrthoFinder ultimately does. It clusters all the proteins that you have that are identified in those proteins into different orthogroups. These are clusters of sequences that are believed to have a shared common ancestor. And as we had hypothesized, the human entry was clustered in a different orthogroup than the Vibrio cholera one. And here you have a tree of the human entry, of the human protein, with a few others. Most of them are paralogs in Homo sapiens, but you also have one of the GNATs in Aspergillus fumigatus in that fungi entry, and clustered with Pseudomonas with a bacterial entry. While in the other group, we have the Vibrio cholera one, the dodecamer, clustered with many different non-swap dimers and hexamers from other bacteria, but also with one protein with that Aspergillus fumigatus entry that was again lonely in between all those bacterial entries, and also with one of the archaeal sequences. And I think this is quite fascinating, because we are now confident in saying that this duplication event that evolves into so many different oligomeric states and acetylation functions already happened in the large universal common ancestor, because this explains how we had one copy, probably already with the capability of domain swapping, that has been preserved in bacteria, in eukarya, where we see is particularly prevalent, especially in Homo sapiens. And in all cases where we see that it domain swaps, we cannot be sure about their archaea presence or absence, because for that we could need to check much deeper all the many more genomes for many more archaeal genomes. And then we had another GNAT copy that doesn't domain swap, that has evolved into other oligomeric states, and that's why when we superimpose them, they look quite alike, like hexamers or dodecamers, that probably has been lost in human. That's why OrthoFinder doesn't cluster any human entry with the Vibrio cholera protein. And that then, coming from completely different genes since the Luca times, the Vibrio cholera and the human entry, and probably among others that we've also seen in this analysis, have evolutionary converts to catalyze exactly the same reaction and binding the same substrate, that is spermidine. And just I would like to finish by acknowledging, of course, Christine Orengo for her guidance, and Nico and Xialin, that now she's doing her PhD in the Max Planck Institute in Germany for kicking off this project, and our great experimental collaborators in the San Francisco State University, Misty Kuhn and Hazel Leiva. Thank you very much, all of you, for your attention.
Thanks for the talk. We have time for questions. Can the next speaker also go close to the stage so we...
Hi, I think you showed a very interesting tree constructive comparing the pocket, and you said you calculated this certain distance, the pairwise distance.
Mm-hmm.
How exactly is this average Grantham distance calculated?
So this is actually an old metric to compare residues, and it's just a chemical comparison of the different atoms among those residues. So you just have 2D metrics with all residues versus all residues, and you get a score. So for example, if you compare isoleucine and leucine, you're going to have a very low Grantham distance, while if you compare a glycine with a tyrosine, you're going to have a very large distance. So what we do is just compare all the residues that we have structurally aligned from pocket to pocket, and we just get the average of those values that we have in that matrix.
Thank you. I would like to ask, when you do the clustering, you just first you cluster with the sequence, and then to choose the representative with the structure and do the next clustering. I don't know whether I understood you right or not, but if you choose the representative cannot stand for the other structure of the rest protein. So how do you think about that?
Yeah, you're completely right. The problem is that what they can experimentally test, it has limits. So what we wanted to find is a way to select the most diverse first sequence. That's why we first cluster at the sequence level with the functional families, and then also structures to get a good representation of the different oligomeric states. Of course, we're missing things out there, and that's why we have, for example, that lonely eukaryotic entry in the top of the tree. Probably there are more there, but just because our selection is biased to the way we did our clustering, we're missing things there. Yeah.
Another question here.
Very nice talk. Thank you. And I have a question. Did you check for other measurements for the binding sites, like such as solvescence, SSO area, or hydrophobicity, total, any other measurement other than just the sequence?
No, not really. For this project, we just focused on the residue conservation. We haven't really, I think, checked on properties like hydrophobicity or electric effect and so on. I guess it can, yeah, for sure it will have an effect, but we didn't get to this sort of analysis.
We have an online question now.
Yes. Ravi Amro is asking, is it possible to speculate why NAT2 might have been lost in humans?
Oh, that's interesting. I guess it's just evolutionary pressure. I guess if for some reason that fault or the function that it was performing was taken over by the domain swap, which we see it's the most common type of polyamine acetyltransferases in human. That's why I guess it was eventually lost. But yeah, it's difficult to, I guess one could check from in a deeper eukaryotic analysis, see from which clade that was, is it still present or not? For example, is it present in monkeys or it's already lost in all mammals and so on? That would be an interesting analysis to do as a follow-up. Thank you.
We don't have much more time for questions. Thank you very much for such a great talk.